Today we're going to be talking about microbial nutrition and growth. Like any organism, microorganisms are also dependent on specific types of nutrients in order to be able to grow well. Macronutrients are those that are required in high amounts, and those include your typical things that you might think about, like carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphorus. These are the major constituents of macromolecules that we are familiar with as large eukaryotic complex organisms, and other types of things that are required for them in large amounts are potassium, calcium, magnesium, and iron. These typically act as enzyme cofactors in metabolic pathways. So, a lot of the metabolism pathways simply do not work unless the enzymes have their affiliated metallic cofactor assigned to them and available. Micronutrients are required in much smaller amounts, could be even as little as trace. And the following are some of those examples. You can have manganese, zinc, cobalt, molybdenum, nickel, or copper. They're often going to be acting as cofactors for enzymes as well in events like nitrogen fixation or in specialized pathways like vitamin B12 synthesis, for example. Other microorganisms depend on these things, but may additionally be able to flex to different types of metabolic pathways, depending on their environment, their morphology, or whatever types of environmental stresses they may be up against. The ability of microorganisms to flip-flop between different types of metabolic pathways when required is something that we see sometimes in larger organisms, but it is especially something that we see in single-celled organisms that are really at the mercy of the world around them. We can classify microorganisms by their nutrient sources, and we've talked about this a little bit already, but based on where they get their carbon, we can call them either heterotrophs, and heterotrophs are going to be getting carbon from organic sources. Within that group, you can have organisms that are considered to be fastidious, which means really that they're picky. They have very specific nutrient requirements and cannot function on a lot of different ones, but really have to have certain things available in order to thrive. Autotrophs are organisms that can fix carbon from carbon dioxide. So often you'll see photoautotrophs because that process is facilitated through light and other types of energy that will allow for carbon dioxide to be fixed and formed into different types of sugars and other nutrients as well. We can also classify them further based on the energy sources that they depend upon or even the electron sources that they depend upon. Like many other organisms, there's always the problem of how do things get inside of the cells. Nutrient uptake in microorganisms is done through some of the very similar mechanisms that we've learned about in the context of eukaryotic cells. We have passive diffusion, facilitated diffusion, active transport, group translocation, and as a specialized one for many different types of microorganisms, iron uptake. And we're going to talk a little bit about each of these in the context of prokaryotic cells now. When we look at passive diffusion, we have to remember that diffusion itself is going to be the movement of molecules from an area of high concentration to one of low concentration. So things that are diffusing are moving down their concentration gradient from high to low. Molecules all have intrinsic kinetic energy that causes them to be in constant motion. This is called Brownian motion, if you've forgotten, and we were able to observe that under the microscope in a lab in first year biology if you've taken that course. One important thing to remember that there is no energy input required for this type of movement. The bigger the concentration gradient that exists, the faster that diffusion will occur. So if you have a ton of molecules that are bumping into one another because of that intrinsic kinetic energy and the consequent Brownian motion, you're going to have molecules pushing themselves away from each other a lot more quickly than you will if you just have a couple of molecules of something. So the higher the concentration gradient, the faster the diffusion. Microorganisms not only have to compensate for the presence of their cell membranes, which as we know are semi-permeable and come with all sorts of different caveats for how things are able to move through, but on top of that also have to deal with the cell wall. So there are even more things that have to happen in order to get things across the cell wall and the cell membrane to the cytoplasmic region where metabolic pathways can do their work. This is an example here of passive diffusion, where on one side of the membrane we have a very high concentration gradient of a molecule, And we have the phospholipid bilayer separating these two environments. We have, in this case, a small molecule that would not be charged, that could pass easily through the membrane to the other side. So, not requiring an additional mechanism like a protein, a transporter, a pore. It can simply pass through the phospholipid bilayer because of its properties. So, things like large molecules will not do this. Things that are charged, like anions or cations, will not do this. This would have to be a relatively hydrophobic molecule that would be compatible with the cell membrane that could pass freely. So what happens is these molecules will start to diffuse because of that motion and bumping into one another. They'll push themselves through this membrane and into the environment where there are less molecules. At every juncture where we talk about diffusion, the goal is always equilibrium, where there would be equal amounts of product on both sides of that membrane so that things are no longer compelled to spread out. When it is spread evenly, you're no longer going to see that change in concentration across the membrane. Things will still be moving, but the concentration is going to be the same across both sides. Facilitated diffusion is another kind of diffusion that requires an additional protein to proceed. This has movement of compounds across a membrane using something called a carrier protein. So this extra little bit of machinery will allow things like larger molecules to pass through. No energy in these cases are required, but what this molecule does is acts as a little passageway with several different conformations depending on how much of product might be found inside of it. So you can see, for example, here on the left-hand side, we've got a couple of molecules that are filling in this protein. And when a certain threshold is met, there's going to be a conformational change, meaning a change in shape, to close off to this side of the environment and open on the other side, as you can see here. So then there's going to be a release of those molecules onto the other side of the membrane. This process does not require any use of energy in the sense that we're not adding energy in the form of ATP, for example. But what we do have is that concentration gradient. So we are harnessing the power of the gradient itself, but no additional energy is being put into this system. Now, the problem becomes when organisms are in an environment where they do not have a concentration gradient to work with to their advantage. So let's say they're in an environment where there are very little nutrients outside of them, but they still need to get stuff into their membranes. Well, how do they work against a concentration gradient? This is done through a process called active transport, where we can move compounds from an area of low concentration to high concentration, but this does require energy. So if you imagine doing work against the grain, you'll always have to add some kind of energy source to allow for that process to proceed. So when we look at the different types of molecules that would be involved in active transport, they're all going to be very specific types of transport proteins for a specific solute. So they're not going to be able to just pick up anything and everything that happens to wash on by and bring it into the cell, but they'll have very specific formations that will only be compatible with certain molecules. There are going to be two main types of transporters. We have primary ones that are going to be ATP binding cassette transporters, or ABC transporters. They use ATP as their primary energy source. Or you can have secondary ones that are going to use energy from ion gradients. So we'll see this in coupled transport processes that we'll talk about in a little bit of detail coming up. When we look at primary active transport, we're going to look at those ABC transporters that I just mentioned. And these are going to bind to a very specific substrate. So again, not just anything, but a specific molecule that they're responsible for moving across the membrane. ABC transporters tend to form a pore, and they'll use ATP as an energy source to move that molecule across the membrane. These can be larger molecules like sugars or amino acids that might be required to break down for energy or to build up new proteins inside the cell. But the key here is that they allow the entry of only one molecule at a time and in a single direction. So this is called uniport, where you have things moving in one direction across the membrane. So here we can see a molecule shown in green that needs to pass to the other side of the membrane. Sometimes these are going to have transporter proteins that will pick them up from the cytoplasm or the extracellular space and bring them to these active transporters as an escort of sorts. And that's what's being shown in this case, but that's not always necessarily going to happen every time. So the molecule will bind to the transporter on one side, and you can see that the transporter is a pore, meaning it is a hole that is controlled, right? It's a protein insert in the membrane that's only going to allow for specific molecules to pass through. Once it binds, we're going to have ATP here that's going to be catalyzed into ADP, and that molecule is going to be permitted to pass through this pore into the cell. So the important part here again is the use of energy moving against a concentration gradient to get things that this cell needs inside. When we look at secondary active transport, there are a couple of different things to think about because usually we're using gradients as an energy source for transport, but these gradients are not diffusion gradients, but rather an ion gradient. So a secondary gradient that's not related to the molecule that we need to move into the cell. For bacteria, these are typically going to be proton gradients. In higher mammals, we'll often see this with sodium or potassium or some other kind of ion, but primarily for prokaryotic cells, this is going to be protons. Proton gradients can be used for a variety of different things, like ATP production by cellular respiration, which you may have seen if you remember the electron transport chain and its complexes that are pumping protons across in order to facilitate several reactions. There's flagellar movement, and then obviously what we're talking about here, secondary active transport. You can have what's called symport. This is when a proton gradient and the solute are transported in the same direction across the cell membrane. So you're having two different types of molecules moving in a single direction. And I'll show you some examples in the next slide. We also have antiport, where we've got substances, so the molecule of interest, and then the proton that are moving oppositely. So one is moving into the cell and one is moving out. But either way, we're using that secondary gradient as an energy source to allow for molecules to pass into the cell when they would not normally be able to do so without help. So here's that example of uniport that we talked about with our ABC transporters. Over here, this is our example of symport. So we've got our ions, right? Our ion gradient, there's tons and tons on the outside, and we want to bring this molecule in as well. So what we're going to have here is in symport, the ions from their gradient, which is going from high to low, moving with the molecule. So they're both moving in the same direction. For antiport, we've got a very high ion concentration out here, like we just established, but there's something inside the cell that needs to get out. So you'll see in this case, the molecule is going to be moving out of the cell while the ion is going to be moving in. Anti is like opposite, so hopefully that'll help you to be able to remember the difference between symport and antiport. This is called coupled transport because, of course, we're using two molecules, right? This one, of course, would not be considered coupled transport because we're moving a single entity across the membrane versus two. This diagram is just showing a couple of different examples of how different types of ion gradients are established. So they're showing here, for example, that in electron transport chain, we're having ions that are getting pumped across the membrane to establish a gradient outside. Same thing can happen with other types of ions, as I mentioned. Here they're showing the example of sodium. And sodium gradients can also be used just as much as any other ion can when we're discussing things like symport or antiport. In this example, sodium is required for this transporter to move something across the membrane. So you'll see that sodium will come in and bind. And you'll see inside of the transporter as well, there's a slot for sodium ions. The sugar that is needed to get into the cell has a secondary binding site inside. When both of these bind, that's going to induce that conformational change. So you'll see that this end is now open, but now this end closes and the other side opens to allow these molecules to leave. So there are a ton of different examples of these. No two are alike, but what is common between them all is that you will have an ion of some kind that will be needed to go either in the same direction or an opposite direction of the molecule of interest to cause some kind of conformational change or harness another gradient's energy to move molecules against a concentration gradient. One thing that's particularly specialized for microorganisms is the requirement for iron, and it is really a common thing that's needed by a lot of bacterial enzymes in order to function. Ferric iron, or Fe3+, is very insoluble and very difficult to transport into the cell. So there are a special type of molecule called siderophores that will bind to iron that is secreted regularly by many different types of bacterial species that can bind iron, and then siderophore receptors exist on the cells that allow for that molecule bound to its iron to be moved into the cell. So these organisms have developed specialized mechanisms that our cells don't necessarily need to allow for required molecules that are difficult to obtain or that don't function like others might, and to get them into the cell for their metabolic purposes. So we'll switch gears now and we'll talk about how we actually grow microorganisms in the lab setting. And to do this, we more or less just need to meet their basic nutritional requirements. Media is whatever type of nutrient source that we provide for microorganisms to grow. It could be liquid in the form of something like broth. It could be semi-solid, or it could be solid. So we've seen in our lab so far a variety of liquid and solid, not so much semi-solid. We will deal with some coming up toward the end of the course, but not until then. The chemical composition can be either defined, where we have a specific set of nutrients that are present in specified amounts, versus something like complex media, where it's just a mishmash. It's never the same twice. We'll talk about some examples. And there's also the functional type. Is it going to be supportive, which is general purpose, enriched to, for example, deal with organisms that might be a little bit more fastidious in nature? Is it selective, where you're trying to only grow certain types of organisms versus all of them? So it might suppress the growth of some, but encourage the growth of others. Or differential, where it can induce a change in the morphology or the color or the shape of a colony in order to help you to identify something. It could be used diagnostically. So growth medium does not only necessarily have to be the nutrient source alone, but can have a variety of other functions as well. These are some examples with some photos of what liquid medium looks like. So we can have liquid here. This is just broth that typically is made with some kind of extract. You can have egg extract, beef extract, anything under the sun that has nutrients in it can be used to grow bacteria. Solid reagents like agar can be used. We've seen these quite a lot in the lab setting. Again, that's that solid jello-like material that bacteria can grow on the surface of. Some bacteria, if they're anaerobic, can be inserted into agar and grow inside of it without the need for oxygen at the surface. Semi-solid are things like gelatin agar. We'll use those later on in the course when we're looking specifically for organisms that digest gelatin as part of their preferred metabolism. But typically, the most common are going to be liquid and solid. For chemical composition, a defined or synthetic media is one where all chemical components are known, especially in the amounts that they are present in as well. So this is for organisms that have very specific requirements for growth, so you can't really push their limits or force them to do things that they're not really comfortable with in terms of being cultured. So you do have to make sure that things are present in the amounts that are required. So you can see this breakdown, and of course you don't have to memorize this. I'm not going to ask you, please name all of the components of BG11 medium for cyanobacteria. This is just an example, but it'll show you here exactly how much, like down to the microgram, what you would need to add to your medium in order to support the growth of these organisms. The same thing down here for E. coli. They're trying to make E. coli seem like they're very picky, but that's very much not the case. E. coli can grow on regular complex media without too much trouble at all. But either way, hopefully this makes some sense. When we look at complex media, this is going to be media where the exact composition of elements is not known. Some might be known, but generally, you don't really know what you're getting. This is typically what we use in the lab. So peptone and meat extracts, we use beef extract a lot, are what mostly go into our agar plates if they are nutrient agar plates. So in these cases, beef extract and yeast extracts are just cells that get dried out, and all of their components might not have the same things that another beef extract might. So they have similar things. You can expect to find the same types of things, but in terms of how much of them are available, it's not necessarily going to be consistent between batches. And again, these are just some examples of recipes for some common complex media. Peptone and beef extract, again, are just powders made from those sources, and you'll add a specific amount of them, but what's present in this 5 grams or this 3 grams will differ again between batches. You can see again for tryptic soy and McConkey agar, there are defined amounts that you're putting in, but all of these things, depending on what batch they were a part of during preparation in a laboratory or a factory, will be different. We'll talk now about the function of certain types of media. There's our supportive media, which we've talked about. General purpose, it'll support a variety of microorganisms to grow. We have our enriched media, which will encourage the growth of those fastidious microorganisms that have requirements for certain things to be present in specific amounts. We have selective media, which is going to favor the growth of a specific organism while suppressing the growth of ones that we are not interested in. And then our differential medium, which can be used to identify microorganisms. So for example, if you were looking for something like fecal contamination in a water source, there's a certain kind of medium that you can grow bacteria on that when these bacteria form colonies, if they are enteric bacteria, or typically found in the bowels, they'll actually develop a metallic sheen. So that would be a great differential medium where you could put a sample onto some agar, allow it to grow up, and then if you do see organisms that do get that metallic sheen, you can confirm, absolutely, that has fecal contamination in it. So they're very useful, as well as allowing us to grow bacteria that are not necessarily going to be happy in normal growth conditions. These are, again, some different types of media, and I will not ask you about these on a test, but this will just kind of show the different mechanisms of action and how these media will allow for certain types of bacteria to grow well, while others are not necessarily going to grow. We will see some of these in the lab, like I mentioned, but for now, this is more just for your information if you're particularly interested in this topic and want a little bit more. Now, we've seen some colonies on some plates throughout the semester so far, and typically what we're looking at with them is a single cell that has been planted on agar, has replicated itself, and grown to large numbers. At the edges of those colonies, if they're large, is where cells are growing the best. They are furthest away from that initial spawn point, so there are likely more nutrients present at the edges. There's also a higher concentration of oxygen at the growing ends, and there's a lower concentration of toxic products or potentially waste products. There's also less cells there, right? If the colony's still growing outward from that center where they originated from, there will be less cells to compete with moving outward. The center of the colony will be filled with microorganisms that are growing more slowly because they're older or potentially even dying. There are some microorganisms that can actually change their morphology or shape based on their environmental conditions. Colobacter species is an example shown here that will change their shape based on phosphorus concentrations. So in phosphorus-rich conditions, they look a little bit straighter. In phosphorus-poor conditions, they tend to look a little bit more elongated and more curved in nature. These changes can be either from stress or some kind of adaptation, depending on what species we're talking about. In order for bacterial cells to divide, they do have to replicate their chromosomes. The chromosomes are typically circular in nature, so we're not going to see a large number of chromosomes like we do in higher organisms. Chromosomal replication is going to begin at the origin of replication, and the replisome, which is made up of a group of proteins, will form there, and the chromosomal information is going to spool through the replisome complex. So the replisome itself is going to be stationary, and the DNA is going to be fed through it to allow for it to be replicated. The replication is going to proceed in both directions from that origin and is going to be completed at the location called the terminus that's directly opposite to the origin. Following that, the next step is partitioning the DNA to cut it apart and push it to either side of the cell as it divides. And that process is not very well understood at this point, but you do have to get the new copies of DNA to either pole as the cell is splitting in order to make sure that each new daughter cell does have its own unique copy of DNA. So here we have a young cell that is in its early phase of its cycle. And as cell division is approaching, of course, we're going to have an elongation of that cell. As we see in cells in us that divide, the cells have to double their membrane. They have to work on their organelles and make more of them. They have to increase the cytoplasmic amount. There's a lot of work that has to be done in order to make two new cells from one. So you do see an increase in size of a dividing cell prior to the division. We also have the replication. You can see this blue DNA that's coming off is going to be the new DNA strand that has been made from the parent DNA. And as these cells elongate and the new DNA gets partitioned, we're going to have the growth of what's called a septum. The septum is going to eventually split these two new cells by binding and forming new complete cell membranes and cell walls between the dividing cells. And eventually they are split apart into two new cells, one with the parent DNA and one with the new DNA. And this is, as we know it, cytokinesis. We have the formation of two new cells in this case. In the cases of cells that grow to form streptoformations or even something like diplo, sometimes the cell division does not break. So you will not see the detachment of two new cells, but they might remain attached in some cases. So this diagram is showing some chromosomal replication. We have our origin up at the top here, and the terminator, as we saw before, at the bottom. And the replisome is going to come and bind at the origin. The origins are going to separate, and that DNA is going to start being fed through and replicated. And over time, we're going to have the elongation of the cell and the production of this new DNA, of course. And then eventually, we're going to have that partitioning, the separation of the two new DNAs into either end of that cell, that septum formation, and eventual splitting to form a new cell, which can then divide further, and so on and so forth. So this diagram is showing E. coli as an example, where we have the initiation mass that's reached, so that cell size is required. Again, that elongation, you're going to have the initiation of DNA replication at that state. That's going to take a very long time. And then in addition to that, you're going to have other factors coming in to allow for the eventual septation step, and then finally the cytokinesis and division of those two newly formed cells. Because every species is different, we can populate what are called growth curves that are used to describe microbial population growth. And again, depending on what kind of organism we're looking at, what the species is, the growth curves will be different. In labs, we tend to grow what are called batch cultures. These are grown in limited nutrients, and there's a high accumulation of waste products. So you make, for example, one Erlenmeyer flask with broth in it, you inoculate it with some culture, and then that's it. Those cells grow up in there, and over time, they're going to divide, but they do eventually get overwhelmed because of the amount of waste products that accumulate over time, the nutrient depletion, and simply the competition with too many other cells to survive for a very, very long time. There tend to be 4 stages of a growth curve. We have the lag phase, the log growth phase, which is considered the exponential phase, the stationary phase, and then senescence and death. And we can see this in any population that is grown in the laboratory setting. So this is what it looks like. We have our lag phase. This is typically after a couple of cells are introduced into a new medium. So growth is going to be pretty slow. And then eventually, once they get acclimated, they start to get some nutrition underway, some metabolic pathways going. They're going to start to divide in large numbers. 2 become 4, 4 become 8, 8 becomes 16, etc. So you see this huge jump in the population until it starts to kind of peter off. The stationary phase kind of gets reached when cells are being born and dying in the same amounts. And then as those nutrient sources deplete, as waste products tend to build up, you're going to see populations slip into the death phase or the senescence phase. And slow but sure, the cell numbers are going to start to come down again. When bacterial species get introduced into new culture medium, that growth is going to be stalled. They are metabolically active, there's nothing the matter with them, but they do need time to acclimate. They're going to have to spend some time getting ready to grow. They're going to need to produce new enzymes, maybe make some new energy, maybe get ready for cell division. So depending on the species, the lag phase will vary in length. Every species is going to have a different amount of time, and even the same type of species cells in different lab conditions might differ depending on what type of environment they're being put into. It can be dependent on the age of the previous culture. Are they old cells? Are they new cells? And also the culture medium itself. What are the nutrient amounts? What are the preferences of the species that we're trying to grow? There's a variety of different parameters that will affect how long that lag phase will take to go away. The exponential growth phase is exactly what it sounds like. We have doubling of cells at specific intervals, following cell division, septation, all the things that we just discussed. Growth tends to be balanced. Cell components are made at constant rates. Cells don't naturally pick up speed at any time, they don't decrease at any time. All cells kind of just go through their motions the same way every single time they're getting ready to divide, as long as conditions are consistent. When the nutritional or the environmental conditions are changed, the growth can become unbalanced in those cases. So if you make any alterations, cells may have to compensate for that or may be affected detrimentally by that change. In those cases, you're going to see the components that are needed for metabolism or cell division being prepared at different rates. Maybe they're slower, maybe they're faster. But the cell division is going to kind of peter off until a new balance is achieved and everything kind of stabilizes over time. When cells are dividing the most happily at their peak, they can reach up to 10 to the 9 cells per milliliter, and that's an awful lot of cells. Population growth in those cases is going to appear to stop because you're going to have cells that are going to continue to divide, but the same number of cells will be dying as well. So you're not going to see the population continue to increase at those points. You can also have cells just stop growing entirely because of things like nutrient shortage or that accumulation of toxic wastes like we discussed. Some cells have survival strategies. They can go into endospore formation mode where they're going to say, okay, well, the environmental conditions are not ideal. I'm going to try to preserve myself in an endospore for a later time when conditions may come back around. But don't forget, not all organisms can make endospores. So this is not something that all species can do. They can start to form starvation proteins, or they can attack their neighbors by increasing their virulence, which will allow them to persist beyond other cells that maybe haven't reached that stress point yet. In some cases, cells will resort to cannibalism. If there is not enough nutrients being supplied through the environment, they will kill their neighbors and eat them. And you can see some examples here of cells that turn into spores. They're kind of saying, okay, well, there's some stress here. It's spore time. They form these spores, but then they also start to secrete factors onto their neighboring cells, and then they destroy them and then eat their insides. So there are lots of mechanisms that protect cells and allow them to maintain and persist in environments that are no longer ideal. If you have cells that are extremely stressed, if you happen to transfer them into new medium where there is tons of nutrients available and they can kind of go back to their happy state again, some of them don't grow right away, or maybe not at all. There are a couple of hypotheses for what occurs in these cases, and one of them is that the cells are viable but not culturable, or VBNC. Cells may just have lost completely their ability to grow. Some starving cells may not be incapable of growing, but they're just unable to for a short time. They've gone into a dormancy state. It could be an endospore. It could be a slowed metabolism. It could be an alternative metabolic pathway that they've conjured up and have to revert back to. But once suitable conditions are met, VBNC cells can snap back to reality and begin to grow again. There are also cases where cells become so extremely stressed that they're going to undergo something called programmed cell death or apoptosis. This is where cells induce cell death because they are unable to survive, so they induce their own destruction from the inside. This usually causes perforation of the cell membrane in the cell wall, which will allow their nutrients to leak out into the environment. It kills them, but it helps out surrounding cells. So some of them can be quite selfless. They can understand that the need for the greater good is to allow for others to persist, even if that doesn't happen to include them. So these are some examples showing what can happen in the death phase. So cells that are in death phase, they're senesced, they are spore forming. What can happen is if you transfer some of these cells to a new plate, some of them will live and snap back and start to grow again. Some of them will undergo genetically programmed cell death in that death phase, where only some of them will survive. right? They can consume the nutrients that leak out of their dead siblings. I love this diagram. In this one, genetically programmed sterility, or those VBNCs that we talked about earlier, where they're just kind of in a dormancy state, they've slowed down, they've changed, they'll take some time, but once introduced into new medium, they will come back to life following that environmental change and can start to grow again. The death phase can take a very long time. If you imagine cells dying a little bit, their neighbors eating them, living for a while, then more of them die and more neighbors eat them, the decline is going to be very slow over time. So you'll see that this can take a long time to get to zero because the cell population can persist for a long time until there's only a single cell left, right? Imagine that that's the case where you're the lone survivor and all of your other cells that you lived and grew up with are gone. So that's how long it can sometimes take. When we look at generation time, this is the number that we allocate to doubling time, or the length of time that it takes for the population to double itself. This is measured during that exponential phase or that growth phase that we talked about. And the formula is 2n, where n is equal to the generation number. We express this growth plot as a log plot because it's an exponential relationship. So you can see in this case, this red line shows cells growing exponentially. At each interval here, they are doubling, but we represent that growth as a log curve because it makes it linear and easier to understand versus the exponential growth curve. When we measure microbial growth, there are a couple of different ways to do this, as we've discussed, but how do you really count bacterial cells when there's so many of them? Well, we can look at cell numbers, where we look at cell divisions in a direct way, or cell mass, where we calculate cell divisions in an indirect way. First, we'll start out with the cell numbers because they're fairly easy to measure, and there are some very interesting direct counting methods that we can use, like the Petrov-Hauser method, the Coulter counter, or even the membrane filter technique. So let's have a look at some of those. When we look at cell counting methods, one that's commonly used is the Petrov-Hauser counting chamber. You can see that this is a specialized slide that you add cells to, and this little spot here has a known depth, so you add on top of it a cover glass, and then what you do is you put the slide under the microscope and then look to see how many cells are present in certain squares on the slide, and then you can use that number to estimate the amount of cells based on that density that you observe. There's also another machine called a counting machine that you can put a sample of bacteria through, and individual cells will be counted as they get passed through an electrical current or light down below. These tools are also useful to count the number of cells that are living or dead in a population, because if you see cells, it doesn't necessarily mean that they're alive. This machine will help to differentiate between living and dead cells based on their shape. You can also do the membrane filter technique, which is going to measure cells from a liquid sample, and one colony is going to be equal to one cell. So what you do is you're going to take a filter and put it into a filtration system. You're going to add your sample to come through, and if cells are present in a specific density, they're going to be transferred from this filter onto an agar plate, and then depending on how many grow up, you can tell what that cell density was, because one colony will be equivalent to a single cell. So that's another way that you can measure how many cells are present in a population. We can also measure the cell mass. We can measure their dry weight, for example. If we were to take a cell culture, centrifuge it in order to collect all the cells, and then take them and put them into an oven to bake off any water, we can physically measure how heavy that specimen is. Or a more common way is to measure what's called the turbidity. Turbidity is how cloudy a specimen looks. If we were to shake up a culture and there's cells that are growing inside, broth starts to look more opaque. Then we use spectrophotometry to see how much light can pass through that sample. So for example, up here, this culture has less cells, those little dots inside, there's fewer of them. So more light can pass through, and we get a different reading on the spectrophotometer, right? Because more light can make it through that sample. When we come down to this example, where there are more cells that are present, this broth is cloudier in nature, less light is going to be able to pass through, which will indicate that there are more cells present. The turbidity amount can correspond to a chart, and based on how turbid or how cloudy your samples are, you can estimate based on that chart how many cells you have in your population. We mentioned earlier that most cultures that we grow bacteria in the lab are finite. We add them to one single container of medium. When all of that is used up, that's it for them. But there is a method called continuous culturing, where we have machines that will constantly introduce the flow of new medium into a vessel and also remove old medium to get rid of waste products to allow for continual, more lifelike conditions for bacteria to grow in. This can be done with chemostats and turbidostats that are going to measure the amounts of cells and also how many nutrients and other chemicals are present to know when to introduce and remove medium. The chemostat is one method here where we'll have an essential nutrient in limiting quantities. So that's going to determine how the growth rate will proceed, because that limiting nutrient will prevent cells from growing too fast and will limit them from growing too slowly as well, depending on how much is added. Fresh medium is added at the same time that old medium is removed, so that will allow for a constant replenishment. Imagine being at a buffet that is constantly being refilled. Other than needing the bathroom once in a while, you're pretty much set. You can stay there forever. So you can imagine this setup similar to that, where we have the old food getting taken away, new food coming in, and you're able to persist there for as long as you like. The turbidostat is something that uses a photocell to measure the turbidity of growing culture. So when I was talking about cloudiness a while ago, you'll see here the tube on the left, you can see through it. So you can see the lines on this card behind it. This other culture that has a lot of growth in it is cloudy, so you're not able to see the lines through. In this case, the flow of fresh media is going to be controlled by how turbid the culture is, and this is going to allow for a maintenance of a consistent cell density. So you're not going to see fluctuations too high or too low. It's going to allow for cells of a known growth rate to maintain those values, to be happier, to not have that buildup of toxic byproducts through waste metabolism and things like that. There's also the distinction between aerobic and anaerobic techniques. When we talk about aerobes versus anaerobes, aerobic organisms grow in the presence of oxygen, where anaerobic ones don't. They like anaerobic conditions or those that are devoid of oxygen. So sometimes we just need to grow bacteria below atmospheric conditions of oxygen that is present in our air. Aerobic bacteria are easier. All we have to do is aerate their medium. So typically we put them into a machine called a shaker. And a shaker is just going to have a platform on it that's going to rotate. And as it rotates with liquid medium on it, that liquid is going to be sloshed around. And at the surface of that liquid, air is going to be able to exchange and penetrate deep into that culture. And over time, that aeration is going to keep enough oxygen present in that culture for growth. For anaerobic media, you have to physically remove oxygen from that environment. So you have compounds that you can add into an airtight container where these organisms might like to grow to suck all the oxygen out and to produce carbon dioxide in higher amounts. You can do this in an anaerobic work chamber. You can see an example of that down here where we've got a gas pack where we're having any oxygen that's free in the air getting converted into water, and we're having the production of carbon dioxide in high amounts. And these are our stack of agar plates in here that are being grown in conditions that are devoid of oxygen. This is important because we do have some relevant species that we need to grow or study that do require anaerobic conditions. So having the means to grow them in the lab is extremely important. And then finally here, if we want to be able to identify microorganisms or diagnose a sample in terms of what it contains, we have to be able to isolate it. It has to be able to be grown in a lab, and then we can use a bunch of different techniques to identify it. This can be quite difficult because if you're just getting a fresh sample in, you might not necessarily know where to start. Do you start with a selective media? Do you start with a supportive media? Do you go anaerobic? Do you go aerobic? There are a ton of questions to ask in terms of what different species might require in order to grow. So working in a microbiology lab has its thrills because you never quite know what you're going to get in a given day, and there's always a puzzle to solve along the way. It's believed that only 1% of microorganisms in our environment have been successfully cultured in a lab setting. So we have a lot of work to do in terms of being able to grow these up in numbers that are large enough to be able to study them, to document them, and to work with them, and obviously to learn more about them as they're discovered. So we'll leave it there for today. And thank you all so much for your attention. Of course, if you do have questions, you know how to reach me. It's laura.r@nipissingU.ca. And I hope I'll hear from some of you soon.